Hello everyone, today we are going to discuss a very important topic of the medical virology, and this is dengue virus. In this short tutorial lecture, we are going to discuss about the history, general characteristics, replicative cycle, epidemiology, signs and symptoms, clinical features, diagnosis and then treatment and prevention of dengue virus. So first of all, we should know that the dengue virus belongs to the family Flaviviridae, and it is transmitted through a specific species, through a specific family of the mosquito, that is Aedes mosquito. Initially this was diagnosed in monkeys, and then about 100 to 200 years ago it jumped in humans and causes severe complications. And the word dengue is derived from African language. Dengue virus is enveloped virus. There is a distinct envelope around the virus, and it is a RNA virus and positive sense single stranded RNA virus. That is why it belongs to the fourth Baltimore group, and there are four serotypes of it. You can see here that this is a schematic diagram of dengue virus. This is the genome, viral genome, that is RNA, and then this is the nucleocapsid, and these are the specific E and M protein that are involved in the adhesion of the virus to the receptor, and this is the viral envelope. The most important thing in this lecture is the replicative cycle. Female Aedes mosquitoes, whenever it bite some infected person, it get dengue virus, and then it transfer it to the healthy individual. There are the most important target of this is the macrophages, and the two main receptors of this dengue virus are the cognate receptors and FC receptors. In most cases, cognate receptors are involved. Whenever it binds with the specific receptors at the surface of the cell and gets into the cell, this is known as receptor mediated endocytosis, and the specific bubble is formed, that is known as endosome. Whenever it gets entry in the cell, it migrate to the rough endoplasmic reticulum. Why it migrate toward the rough endoplasmic reticulum? Because as it is a positive sense single stranded RNA virus, it can directly translate its genetic material to form its specific protein. So that's why it moved toward the RER to get some replicative, to get some machinery, protein machinery, that is ribosomes, to make the its specific proteins. So whenever it it fuse with the ribosomes that are present on the surface of rough endoplasmic reticulum, and then the translation is performed, and specific replication protein complexes are formed that are used to manufacture or you can say that replicate the specific genome of the dengue virus. The positive sense strand is firstly set as a template strand, and negative sense strand is formed. Then this newly formed negative sense strand is taken as template, and identical copies of positive sense strand are created. And then after the multiple copies formation of the genome of dengue virus, it migrate the viral genome, and some viral protein migrate toward the Golgi complex for the maturation purpose. There is a specific pH that is involved in the maturation of this virion particles, and then after maturation this is exocytosed. You can see here that this is the entry, and then the uncoating, and then this is the and this is a translation process of genome of the virus, and then it is again the fusion, and then in the RER after in Golgi complex, and then exocytosis of the matured virions. As well as epidemiology of the dengue virus is concerned, about 400 million cases are reported every year, and about 22,000 deaths occur due to this dengue virus. Mosquito is the main cause of it, that is basically involved in the transmission of it. There are some signs and symptoms like fever, abdominal pain, nausea, vomiting, pain behind eyes, rash, muscle joint pain, and headache, swollen glands. Whenever a patient is whenever a patient is presented in a clinic, there are some clinical features. There are leukopenia, basically the low level of white blood cell, mucosal bleeding, liver enlargement, thrombocytopenia, the decreased level of platelets, persistent vomiting, positive tourniquet test. Diagnose. How you can diagnose the dengue virus? You can diagnose the dengue virus by using direct and indirect diagnostic techniques. Direct techniques like PCR, tissue or cell culture, and the indirect techniques like antibodies and so many other are there like ELISA, RIA, radioimmunoassay, etc. There is no specific treatment for the dengue virus. Supportive medicine like antipyretic fluids are administered, paracetamol, etc. Why platelet count decrease in dengue fever? The most important question. Some viral proteins directly target bone marrow and reduce the platelet production. As you know that bone marrow is the factory in which different blood cells are manufactured. So whenever viral protein directly target the bone marrow, it it reduce the platelet production, and some immune responses of dengue infection can also decrease the platelet count. And in chronic conditions, you can see that the plasma leakage can also cause the low platelet count, and some virus can directly cause platelet destruction. So this was all about the dengue virus. Thank you so much. If you have any question, then leave in comment. Thank you so much.